In April 2021, a government scientist crouched down in a residential backyard in the Florida Keys and placed a small cardboard box on the ground. Inside the box were mosquito eggs, not ordinary mosquito eggs. These had been engineered in a laboratory. Their DNA had been rewritten. A gene had been inserted into every single one of them, a gene that had never existed in nature before. Designed by a British biotechnology company called Oxitec, tested 4 years in contained facilities, and now, for the very first time in the history of the United States, being released into the open air. The box was left in place. The eggs hatched, the mosquitoes flew out, and within weeks, they were mating with wild mosquitoes across the Florida Keys, carrying their engineered gene into a natural ecosystem that had never seen anything like it before. This was happening in 6 locations simultaneously. Then more. Then more. Over the following 3 years, hundreds of millions of genetically modified mosquitoes would be released across the Keys. The EPA approved the experiment. The state of Florida approved the experiment. The Florida Keys Mosquito Control District approved the experiment. The plan was then expanded to potentially cover 2.4 billion mosquitoes across 4 counties in California. Billions. With a bee genetically modified. Released into the wild in the United States of America. And here is the part that the headlines at the time mostly missed. The part that makes this story genuinely extraordinary. The answer to the question that almost nobody asked clearly enough at the time. What actually happened. Not what Oxitec said would happen. Not what the protesters said would happen. Not the press release, and not the petition. What actually, verifiably, measurably happened when you release billions of engineered mosquitoes into a real ecosystem. With real weather and real wild mosquitoes and real human beings living their lives around them. That is what this video is about. To understand why Florida did something this radical, you need to understand the enemy, the yellow fever mosquito. It is not native to the United States. It arrived from Africa centuries ago, carried in the water barrels of ships, and it has been spreading ever since. It is not the mosquito that most Americans think of when they think of mosquitoes. The big, buzzing, bite you on the arm at a barbecue nuisance. Aedes aegypti is smaller, stealthier. It prefers to bite indoors. It bites ankles and elbows, rather than the obvious exposed skin that most mosquito repellent is applied to. And it carries diseases that the mosquitoes you grew up with do not. Dengue fever, Zika, Mosquitoes, yellow fever, chikungunya. These are not minor illnesses. Dengue kills around 40,000 people globally every year. Zika, during its 2015 to 2016 outbreak in the Americas, caused a wave of severe birth defects. Babies born with abnormally small heads, with brain damage, with lifelong disabilities that terrified public health officials across 2 continents. Yellow fever is a hemorrhagic virus that has killed millions throughout history. Chikungunya causes joint pain so severe that patients describe feeling as though their bones are breaking from the inside. In the Florida Keys, Aedes aegypti makes up just 4% of the total mosquito population. 4%. But that 4% is responsible for 100% of the mosquito-borne disease transmission in the area. Between 2009 and 2010, 88 people in Key West contracted locally transmitted dengue fever, the first locally transmitted cases in Florida since 1934. In 2020, another 72 people were infected. Each case had to be traced, contained, and managed at enormous public health cost. In an area where dengue had been essentially absent for 3 generations, and the existing tools for controlling Aedes aegypti were failing. Insecticides, the primary weapon of every mosquito control district in the country, were becoming less effective. The mosquitoes were developing resistance. The chemicals that had worked for decades were working less well every year, and the public, increasingly aware of what broad-spectrum insecticide spraying does to other insects, to butterflies, to the broader food chain, was becoming less tolerant of it. The Florida Keys Mosquito Control District had been looking for a different answer for years. In 2009, after the dengue outbreak, they reached out to a small British biotechnology company that had been working on something genuinely new. Oxitec. Oxitec was founded in 2002 at the University of Oxford. Its core idea was elegant in the way that the most dangerous ideas often are elegant, logical, and completely without precedent in the natural world. What if you could make mosquitoes kill themselves? Not with poison, not with traps, not with chemicals sprayed from planes, What if you could engineer a mosquito so that its own offspring, when bred with wild mosquitoes, carried a gene that killed the females before they could grow up, mate, and bite? Here is how it works. Male mosquitoes do not bite. Only females bite, because only females need blood to develop eggs. So if you could flood a wild mosquito population with engineered males that carry a self-limiting gene, a gene that causes female offspring to die before reaching adulthood, you would progressively reduce the proportion of females in the population. Fewer females means less biting. Less biting means less disease. And because the engineered gene is self-limiting, designed to fade out over a few generations, it does not persist indefinitely in the ecosystem. That was the theory. It had worked in Brazil. In the Cayman Islands, Oxitec trials reduced the target mosquito population by 95%. In Wazzetto, population reduction reached 80 to 95%. The science was compelling, but this was the United States. And the United States had never allowed anything like this before. It took more than a decade to get here. The Florida Keys Mosquito Control District first approached Oxitec in 2009. The regulatory process through the EPA, the Florida Department of Agriculture, public comment periods, and interrogancy review took 11 years. Residents were deeply divided. Some were enthusiastic. Dengue had frightened them, and a chemical-free solution was appealing. Others were furious, calling it a Jurassic Park scenario and threatening to fill release boxers with bleach. A 2016 referendum showed 31 of 33 precincts in favor. In May 2020, the EPA approved the permit. In April 2021, the first boxers went into the ground. The actual numbers were far smaller than headlines suggested. The initial trial released roughly 144,000 mosquitoes across 6 locations. A second phase aimed for 20 million. The billion figure came from a separate California expansion, approved in 2022, not from Florida. The numbers were smaller than the headlines said. The precedent was exactly as large as it sounded. Phase 1 concluded in 2022. Results were a success in carefully defined terms. The engineered males behaved like wild males, competed for mates, and passed on the self-limiting gene. Female offspring that inherited it died before adulthood. The gene disappeared naturally within a few generations. No armed tubies butterflies or other non-target species was documented. The EPA extended Oxitec's permit and approved the California expansion. By summer 2024, the Florida release phase had formally concluded. But here is where the story gets complicated. The trial was not designed to measure what most people actually want to know. Did dengue cases go down? Did the mosquito population drop enough to show a real public health impact? The answer is, we do not fully know yet. The trial was a feasibility study, confirming the technology worked in open air, not measuring disease reduction. Critics noted the data was never independently verified before the EPA approved expansion. A 2021 peer-reviewed study also found that some hybrid offspring in a Brazilian trial survived longer than expected, raising questions about whether engineered DNA could persist unexpectedly in wild populations. Oxitec and the EPA reviewed it and found no safety concern. Critics disagreed. The technology works as designed. The safety record is clean. The public health impact is still being measured. Unlike other ecological disasters in this series, mouse sparrows, Australia's emus, Escobar's hippos, this story does not have an ending yet. What it has is a direction. The tropical disease frontier is moving northward. In 2023, Florida recorded its first locally transmitted malaria cases in 20 years. The old tools are failing. Something new is needed. The United States has crossed a threshold it cannot uncross. Engineered DNA has moved through wild mosquito populations. No disaster followed, but the experiment is not over. Florida, unlike the others, tried something genuinely different, working with the biology of the mosquito rather than against it, targeting one invasive species with surgical precision while leaving everything else untouched. Whether that proves to be a breakthrough or produces an unforeseen consequence is a question only time can answer. It started with a cardboard box in a Florida Keys backyard, a scientist crouching down. A decision nobody could fully take back. The EPA is still deciding. The data is still coming in. Was this brave, reckless, or both? That question belongs to everyone. Tell us in the comments, subscribe, and come back next week, because the true stories are never finished.